The first, well, this is interphase, but the first and fourth phases are the busiest, right? A lot going on to where you can't draw it all happening. But what's happening is the chromatin is eventually condensed into the X-shaped chromosomes. The nuclear membrane is going away. The spindle's getting built between the centrioles, and the chromosomes start to migrate toward that line they're going to form down the middle. Once they've formed that line, we have metaphase. Metaphase also including the spindle fibers, which of course are made of microtubules attaching to each centromere of each chromosome. Kind of glossed over it yesterday, but the spindle fibers that grow the wrong way, I drew some in here just so we have some continuity. Those are called asters, just kind of a side thing. As soon as you see them separate, it's anaphase. They separate and the little crease starts to form at the top and bottom of the cell. The crease gets deeper, the chromatids move farther apart, and the nuclear envelopes reform in telophase, as well as the chromatids actually uncoiling and turning back into chromatin eventually. Now, this is different depending on whether we've got a plant or an animal cell. I drew this in later in the day to show that if this is a plant cell, because there's a cell wall and because the cell is kind of regularly shaped, kind of brick-like and not flexible, what happens instead of the pinching in is that kind of just initially just sort of, you'll see just sort of a hairline crack form, just a real faint line. And then eventually that gets built into a solid cell wall and the new cell membranes on either side of it form. That's plants, right? Animals can do the pinching in part, and microfilaments because they're flexible. Plants, not so much. But eventually both of them wind up being separate cells, and that is called cytokinesis. A little different in animals versus plants. Questions about any of that? Like anything, this is different in the real world than it is in a little cartoon we draw, right? And so the goal now is to get you to be able to recognize real cells in the different phases of mitosis. We are still, policy-wise, a little gun-shy about microscopes at this point. So we're going to kind of do an alternate version of this, starting with you hunting down some photos on the internet on your own of the different phases of mitosis. Basically, your job today is going to be to build a slideshow of your own, a Google Slides presentation. This has already dropped into Classroom, so you don't have to write all this down. It's in there. Basically, a slide for each phase of mitosis plus one for interphase, where you've got a photo of each of an animal and a plant cell caught in that phase. That's the idea today. Let me see if my videos work here. I think. Because just to give you a sense of how messy this can be, is this what I want? Oh yeah, this is cool. You like this. We talk about chromatin winding up into chromosomes. Here's a look at what that actually looks like. So here's a strand of DNA. That's a protein called histone. And what happens is this thing binds around histones. And we get this first level of coiling. Now we're up to a second level of coiling, and the little beads kind of coil together. There's two. Now we call it chromatin. Even chromatin isn't completely unbound. Now we see a third level of coiling and a fourth level of coiling. And finally, a fifth level, and that's what chromosomes are. And so now we bleed from cartoon into real life, and here's a human cell actually doing mitosis. This is about metaphase. anaphase, telophase. It's a mess, right? Chromosomes are huge. They're not little X shapes that we can draw neatly. They're big and floppy and kind of messy, and that's what it ends up being. And so as you get used to what these look like in photographs, which is what you're doing today, you'll get to the point where you can recognize them. And by the end of tomorrow, you'll have something where you can kind of quiz yourself and get to where you can recognize them all. But so baby steps here. What you want today for tomorrow is basically a slideshow with dual pictures of that phase in a plant and an animal cell and a description of what happens during the phase. Okay, just read the requirements on the slideshow, and I think it'll kind of make sense to you. Go ahead and go into the classroom and find it. And you have the period to work on it. That's why it's due tomorrow, because you should have plenty of time to finish it.